So during the conference, you've given a really interesting talk on the connection between stem cells or pluripotent cells and aging. So what's the major link? Does it mostly have to do with the fact that we lose pluripotent cells as we get older?
Yes, so that's an interesting question, and it is true that as human and model organisms like mice grow old, there is a decline in a number of adult stem cell, and in particular in adult stem cell function. So the number of cells stays pretty equal, but their ability to give rise, to proliferate and to give rise to progeny is declining during aging. So there is this relationship basically between the decline in the stem cell pool and the decline in the physiology of the tissue. So if we take the brain as an example, if there is a correlation between the decline in neurostem cell reservoir and cognitive function. And an important question is whether the decline in neurostem cell pool underlies, at least in part, the decline in cognitive function.
Do you think that one day pluripotent cells will offer a solution to some of the most damaging effects of aging? And for what conditions or diseases do you see the most promise in this area?
Yeah, so this is another very interesting question in the sense, and it has two aspects to it. So one first aspect is that we all have, as humans, those reservoir of stem cells. And as I said, their number is not really declining. It's really their function that's declining. So if one were to find ways to reactivate or rejuvenate those dormant pools of stem cells in an old organism, one could tap into the potential or harness the potential, the regenerative potential of those cells. So that's one way in which they can be seen as potentially interesting for diseases. And then the other aspect is obviously like stem cell therapies, whereby they would be grown in a dish and then injected in different sites in the body and take their place and in their new location generate new differentiated cells and help to regenerate the tissue that would be damaged. So in terms of specific diseases, there is already a clinical trial actually in California with Geron, where they are using stem cells and in particular oligoprogenitors for spinal cord injury. As far as diseases of aging, because most of those diseases or a lot of those diseases, in particular in the brain, are degenerative, one could imagine that for Alzheimer's or Parkinson's disease, the ability to possibly reimplant stem cells that have the ability to differentiate into neurons could be helpful. But one has to be very cautious because they need to integrate in the right place. One has to be sure that they don't carry mutations or the ability to form brain tumors. So far it's not clear yet how helpful practically this will be, but on a theoretical basis, it holds great promise, that's for sure.
You're a European scientist who's working in the United States now. How do you think Germany and Europe stack up in this area of research?
Yeah, so there are amazing stem cell scientists in Germany, that's for sure, and I could see that, I knew that already, and I could see that even more at this meeting. Maybe the difference is in the numbers, obviously. So in the US, there are those places, like California is a good example where we are, like where the state of California has put a state effort basically towards stem cell called California Institute of Regenerative Medicine, or CIRM, where they have billions of dollars or like millions of billions of dollars that are geared towards stem cell research.
What topics or themes that have been addressed at this conference do you find the most interesting?
Yeah, that's a tough question. Everything is interesting, I find. So I've learned a lot because my field is aging. It's not really stem cells, so we study stem cells as it pertains to aging. So being in a pure quote-unquote stem cell meeting is very exciting because I get to learn a lot. So I get to learn about reprogramming of cells, whether back to the stem cells or forward into like specific tissues, and I think that's interesting. I get to hear about organ formation, like full organ formation, like we had a talk today on that by Ken Zarrett. I thought this was remarkably interesting. One gets to hear also about the epigenetic changes that regulate cell fate or stem cell function, and I think that's particularly an interesting field as well. So yeah, it's like being in a candy store, a kid in a candy store. You hear about many, many interesting topics.
When this conference takes place again in the next 5 or 10 years, do you think researchers will be mostly focused on the same fundamental questions, or do you think there'll be a shift? Do you think we'll be a lot closer to potential therapeutic applications of the cells?
Yeah, so I think it will be completely a different landscape in 5 or 10 years from now, because if one takes past history as what will predict the future, in 5 years there's been like several revolutions in this field. So that one could not have been predicting before, like the ability, for example, to reprogram a cell back into a stem cell stage, the ability to transdifferentiate one cell type into another, the remarkable identification of the factors that are important, etc. This has been really a fantastic 5 years that I think nobody could have predicted that it would be so revolutionary. So and also because there's so many people in this fast-moving field, this helps, in fact, the field being exponential, in fact. So I would predict that in 5 or 10 years, it's going to be, a completely different landscape. Are we going to be closer to therapies? I really hope so, because today at the meeting it was clear that people are now getting closer and closer to being able to have cells that really resemble, that are made in vitro, but really resemble their counterpart in vivo and really create even the structure of the tissue. So I think this looks remarkably promising.